Okay, welcome to the video on glycolysis and anaerobic respiration. Everything you need to know, or at least some of what you need to know. Hopefully you'll get at least the basics hammered. So if you have a piece of paper to hand, that'll help you a lot. You can draw it out. I'm going to be using this piece of paper to explain what is happening. So glycolysis and anaerobic respiration. Well, whether oxygen is present or not, glycolysis is always the first step in respiration. It takes place in the cytoplasm of a cell, not in the mitochondria. It is important that you understand that. It is in the cytoplasm, not the mitochondria. Let me just get this into focus. We start off with a molecule of every cell's favorite respiratory substrate, glucose. And glucose has got 6 carbons. Now I know glucose is normally shown as a ring. It is a carbon ring. I'm just going to show you 6 in our straight line to help you remember that there's 6 carbons present. Now the first step in glycolysis is the breakdown of glucose into 2 3-carbon intermediaries. So what I'm going to do is I'm going to take this molecule of glucose and I'm going to split it in 2. Like so. In order to do this, we need to use ATP. We need to use 2 molecules of ATP, in fact. And what we're doing is we're phosphorylating the glucose molecule in order to make it more reactive. So this is a process called phosphorylation. Like so. Now we produce 2 3-carbon intermediaries, and it is important from this point on that you remember that everything is doubled. You don't need to know the names of those intermediary compounds. If they are on AP exam, then almost certainly they will give you the names if you need them. So these 3-carbon compounds are then transformed into another 3-carbon compound called pyruvate. And remember, this is happening to both of these molecules. So from one glucose molecule, we get 2 pyruvate molecules. Now, the nice thing about this step is it releases energy, which is great, because it means that we're able to get things done. It releases it in 2 forms. One is the conversion of NAD plus to NADH. NADH is a high energy electron carrier that will move to the electron transport chain inside a mitochondria and provide us with a large amount of ATP. As well as that, we have something called substrate level phosphorylation. This is phosphorylation that's taking place as a result of a chemical reaction in the substrate. 2 ADP are being converted into 2 ATP. Adenosine diphosphate into adenosine triphosphate. Remember, ATP is the energy carrier for the cell. It's the cell's preferred energy currency. It's important to remember that everything that's going on over here is of course going to happen over on this side too. So I need to add in NAD plus being converted to NADH. And I also need to add in 2 ADP being converted to 2 ATP. And the same things will happen to those molecules. Now, in aerobic conditions, those are conditions with oxygen, the following will happen. I show you the edge of a mitochondrion here, and I show you, remember that of course it has 2 membranes, and the inner one is folded into these structures called cristae. The pyruvate will move into the matrix, that is the space within the inner membrane, like the cytoplasm of a mitochondria, where it'll take part in Krebs cycle, also known as the citric acid cycle. The NADHs will move to the electron transport chain, which is located on the inner membrane of the mitochondria. And there they can help to generate more ATP. I'm just going to abbreviate that to ETC. That stands for the electron transport chain. Okay. Zoom back in. Okay, so this is what happens in the presence of oxygen. The ATP can go off into the cell and be used for energy. The NADH will go to the electron transport chain to make more ATP, and the pyruvate will go to Krebs cycle, which in turn will create more high energy electron carriers and ATP, which the cell can use as an energy supply. It's important when you're studying glycolysis that you understand what is being produced. So let's have a look at this. 2 ATP were used at the start, but I have a total of 4 ATP being produced at the end. So glycolysis produces a net total, and this is, I'm just talking about glycolysis alone here, of 2 ATP and 2, if I look back, NADHs. Which of course are going to be converted into more ATP. Now this is what happens when oxygen is present in aerobic respiration. But in anaerobic respiration, we have a series of problems. Essentially, the electron transport chain will stop and Krebs cycle will stop. I'll explain why in another video, but these two processes stop. This results in a buildup of pyruvate, which would be fine for your cells, apart from the fact that this NAD plus is not being produced. Normally it would be released by the electron transport chain and would come back out to here so that glycolysis could continue. So we need to make sure when we are respiring anaerobically that this process at least is able to continue, that we are able to make this small amount of ATP and to continue making it. So the whole purpose of anaerobic respiration is to keep making ATP, and in order to do that, we need fermentation. Now there are 2 types of fermentation. There is fermentation in animals and in some bacteria. They also do the same thing, just a few of them. And over the other side we have plants and fungi. And there is one fungus in particular that they will always love to test you on, that is yeast. Because that's the one we use to make alcoholic drinks. So of course they're going to test you on it. So what is the purpose of this? Oh, my Zoom is playing up, hang on one moment. The reason we want to keep this going is to produce ATP. And in order to keep it going, we need to make more NAD+. So we're going to sacrifice our NADH and convert it back to NAD+. In order to do that, we need our pyruvate. So I'm going to start with animals, I think, because they're a little more straightforward. Pyruvate is converted into the substance that burns your muscles when you exercise hard, lactic acid. You may see in an exam that that's written down as lactate. If it is, don't freak out. Same stuff. Remember, the whole point of this conversion to pyruvate, from pyruvate to lactic acid, is to regenerate the NAD+. So we take in the NADH, and the NAD+ is produced. This is a reduction reaction because the NADH is giving its electrons to the pyruvate. In plants, lactic acid is not produced. Instead, we have two different compounds produced from the pyruvate. Ethanol and carbon dioxide. Ethanol and carbon dioxide. Notice that there wasn't any carbon dioxide produced in animals when they were respiring anaerobically. Again, in plants and fungi, this has the same purpose, to convert NADH back to NAD+, so that glycolysis can keep happening. It's really important. I'm just going to give you a couple of little extra bits of information that you may need at some point. They will be cycled over at some other point in the year, so don't stress too much if you haven't got them now. But a couple of notes. Remember, anaerobic respiration is emergency respiration in order to provide that little bit more ATP that we need to escape from a bear or win some competition. But we pay for this lactic acid in that we have to break it down using oxygen. This creates something called oxygen debt. The amount of oxygen that is needed to break down the lactic acid that is built up in our system as a result of respiring anaerobically. It's part of the reason why we have to breathe heavily after exercise. This particular reaction was one of the first that was written down by man ever. The reason for that is ethanol is this is how we make alcoholic drinks. We force yeast to respire anaerobically by placing it in anaerobic conditions. Often that'll be a container with a sealed lid, but it's important to have some way for the carbon dioxide to escape. So often there'll be some kind of valve set up so that as the pressure builds up inside here, as carbon dioxide is produced, the carbon dioxide can escape, but oxygen can't get in. If oxygen isn't able to get in, the yeast inside will not produce any ethanol. So when they're making alcoholic drinks, it's very important that they maintain anaerobic conditions. So that is essentially everything you need on glycolysis and anaerobic respiration. If you have any more questions, just see me about it. But remember, anaerobic respiration is making 2 molecules of ATP. That is a net total because we needed 2 molecules to start and we're making 4 at the end. Have a nice day.